I'm Brian Jones. I was head of clinical immunology at Queen Mary Hospital for 30 years and taught basic immunology to medical students at the University of Hong Kong. I have always been interested in balance between the different arms of the immune system and how it can be maintained to ensure good health. I'd like to show you some slides which I hope will help you understand some of the complexities of immunological protection from disease. The first half of the talk is on basic cellular immunology, trying to keep it as simple as possible, but not going into some of the more complex genetic pathways which induce immune cell activation. The second half deals with effects of aging, stress, exercise, nutrition, and modern Western living on immunity. The immune system has the job of protecting us from two types of threat. Those from outside the body, i.e. the myriad pathogenic infectious microorganisms in our environment, and from within, the daily occurrence of millions of slight errors in DNA which occur during cell division, which could potentially lead to malignancy. The immune system for most of us performs these jobs smoothly and efficiently, but inborn or acquired immunodeficiency can lead to overwhelming infections and widespread cancer. All the cells of the immune system are derived from stem cells in the bone marrow. These include cells of oxygen transport, clotting, and the cells we will be interested in, the white blood cells or leukocytes. Phagocytic cells responsible for engulfing foreign particles and bits of dead cells from host tissues undergoing repair include polymorphs, macrophages, and dendritic cells. Macrophages and dendritic cells are also responsible for processing the material they engulf into smaller fragments and presenting them on their surface in a form that can be recognized. These cells also produce a range of hormone-like molecules called cytokines, including interleukins 1, 6, 10, 12, and 18, tumor necrosis factor alpha and beta, transforming growth factor, and other growth factors for lymphocytes and phagocytes. We will talk more about cytokines later. The cells responsible for antigen-specific responses are the lymphocytes, of which there are three main lineages. T cells are so called because they develop and undergo selection for reactivity with foreign antigens in the thymus. They are responsible for killing target cells, immune regulation, and production of cytokines, which are soluble mediators of all the different pathways of the immune response. NK, or natural killer cells, are an early form of defense against aberrant cells that need to be destroyed quickly, like virus-infected cells and cancer cells. B cells develop mainly in the bone marrow and are responsible for producing circulating antibodies, which neutralize toxins and activate other defensive mechanisms. Immunity can be divided into innate and acquired or adaptive systems. In addition to the first-line defenses of skin and mucous barriers and the removal of particles by the hair-like structures of the upper airways, the innate system springs into action whenever a threat is recognized by phagocytes through structures on their surface called threat receptors. Most pathogens have molecules on their surfaces that bind to the threat receptors and initiate the processes that lead to activation of adaptive immunity. These include production of cytokines that attract cells that can directly kill the invaders and induce an inflammatory response against them. The phagocytes also process the ingested material into forms that can be recognized by antigen-specific lymphocytes in adaptive immune reactions. The invader, such as a bacterium, with various surface antigens foreign to the host, is engulfed by the surface of the phagocyte flowing around it. This creates a cytoplasmic vesicle called a phagolysosome. Here the proteins are digested into small antigenic peptides. These bind to the histocompatibility locus antigens, or HLAs, that coat the vesicle endothelium. HLAs are responsible for transplants being rejected, but their principal purpose is in antigen recognition by T cells, as we shall see later. HLA peptide complexes are then exported to the cell surface for stimulation of T cells. Every lymphocyte has a unique antigen receptor. The B lymphocyte receptor, or BCR, is made up of the antibody or immunoglobulin that will eventually be secreted into the circulation to coat and inactivate the threatening molecule. Immunoglobulins have a Y-shaped structure made up of 2 heavy chains and 2 light chains. These combine to produce the 5 classes of antibody: IgM, IgG, IgA, IgD, and IgE, each with a terminal so-called variable region into which antigen fits like a key into a lock. IgM is the first antibody class produced in response to invasion, and it can agglutinate invading cells and activate an enzyme cascade called complement, which causes phagocyte accumulation and killing of target cells. IgG is the most important high-affinity circulating antibody. It can bind and neutralize soluble toxins released by microorganisms, and when bound to antigen, it can activate the complement system and attract phagocytes. These cells can bind to IgG-coated microorganisms to initiate ingestion. NK cells can also recognize IgG-coated antigens and take part in IgG-dependent cell killing. IgA is the main antibody class in secretions and is protected from digestion by catabolic enzymes in saliva, urine, and the gut by a bound so-called secretory component. IgD is present in very low concentration in the circulation and is mainly found on the surface of immature B lymphocytes as an antigen receptor. IgE is important in destroying parasitic worms, especially helminths, and is the main class of antibody to cause allergies, asthma, and anaphylaxis. T lymphocyte receptors, TCRs, are also made up of 2 chains consisting of constant and variable regions, the latter binding to specific antigenic peptide in complex with the HLA molecule. There are other peptide chains not shown which participate in the binding of the TCR to the antigen-presenting cell and for transmitting a stimulatory signal into the T cell. It is important to understand that unless lymphocyte receptors that react with normal body components are removed or inactivated, the immune system will attack and damage tissues and cells. When immature cells of the developing immune system attach to self-antigens in the absence of a second signal, they will be stimulated to commit suicide rather than respond in a process called programmed cell death, or apoptosis. This leads to the deletion of most of the specificities towards self-antigens. But if this regulatory mechanism leaks, anti-self T cells could survive and later initiate autoimmune disease. Thus, the mature immune system should mainly have lymphocytes with receptors able to recognize foreign, threatening antigens entering the body, with additional regulatory mechanisms for dealing with any aberrations. Mature lymphocytes will also have developed the surface receptors for the second signal, a molecule called CD28, which must bind to CD80 or CD86 on the antigen-presenting cell for activation and proliferation to take place. When the T cell receptor is engaged by peptide HLA on the antigen-presenting cell and signal 2 is received, rapid cell proliferation begins. This requires further signals derived from cytokines such as interleukin-2, also known as T cell growth factor. After several cycles of cell division, a clone of identical cells is produced to attack the invader. Now, if nothing were done to regulate clonal selection, masses of cells would remain and fatal tissue damage would result. Instead, the system is closely regulated so that once the threat has been neutralized, the majority of cells are programmed to die by apoptosis. However, a few of the progeny cells, which have undergone a process of maturation to develop antigen receptors of high affinity, survive and remain as memory cells, quiescent within the lymphoid tissue. These can respond rapidly and strongly if there is a subsequent exposure to the same specific antigen that caused the first activation. Unlike T and B lymphocytes, NK cells do not have antigen-specific receptors, and this makes them much more part of innate rather than adaptive immunity, though they are influenced by antibodies and cytokines produced in antigen-specific responses, which increase their killing effectiveness. They have two important receptors. The receptors for potential threats recognizes cell membrane components, which induce an activating or on signal. The second receptor recognizes HLA molecules and switches off the activation process. Thus, normal cells are protected from NK killing by the negative signal, while cancer and virus-infected cells often have reduced expression of surface HLAs, so the NK cells do not get the off command and proceed to destroy the threat. T lymphocytes can be further divided into subsets based on the expression of various surface molecules known as cluster of differentiation, or CD antigens, and the cytokines they secrete when stimulated. CD17 positive inflammatory T cells secrete interleukin 17, IL-21 and IL-22, which induce further mediators that attract polymorphs and macrophages. CD8 positive cytotoxic T cells can be activated by specific antigen on the surface of target cells to secrete molecules called perforins, which drill holes in the cell membrane. They also secrete tumor necrosis factor alpha, interferon gamma, and cytotoxic granules that can destroy the target cell. CD4 T cells are crucial to all arms of the adaptive immune system, as they help or regulate all other immune pathways. CD4 positive T regulator cells are important as they downregulate immune reactivity once any threat has been overcome. They do this by secreting inhibitory cytokines interleukin 10 and transforming growth factor beta, and by cell-to-cell interactions. CD4 positive T helper cells are further divided into Th1 and Th2 cells, depending on the type of immune response they induce. Interleukin 12, secreted by dendritic cells and macrophages, is critical in inducing Th1 cells, while if there is no IL-12, CD4 T cells can become Th2. Importantly, cytokines produced by Th1 cells suppress Th2 induction, and vice versa. Th1 cytokines include interferon gamma, tumor necrosis factor alpha, and interleukin 12, which increase activity of phagocytic cells and cytotoxic NK and CD8 positive T cells in the cell-mediated immune response, while Th2 cytokines, IL-4, IL-5, and IL-13, drive the activation of B cells and the production of antibodies. Now we come to my main message: how a healthy immune system, and indeed health itself, relies on a well-balanced immune system. The first type of balance I want to mention is that between Th1 and Th2. Th1 cells are responsible for inducing cell-mediated immunity for killing cancer and virus-infected cells, and while the Th1 reaction is going, Th2 cytokines are suppressed. Th2 cells, on the other hand, induce antibodies which neutralize extracellular pathogens and the toxins they produce, and similarly, an ongoing Th2 reaction suppresses Th1. Once the particular threat is resolved, normal balance must be restored. Failure to downregulate Th1 can lead to autoimmune diseases like rheumatoid arthritis and thyroiditis. On the other hand, prolonged Th2 reactivity can occur in cancer and chronic infections due to the suppressed Th1. Type 2 cytokines are strongly involved in IgE production and therefore are responsible for the causation of allergic disease. Another important balance is that between activated immune cells causing inflammation of the tissues and its control by T regulator cells. Lack of regulation is a major cause of allergy and autoimmunity. Too much downregulation clearly leaves the body in an immunodeficient state and liable to chronic infections and malignancy. At the time of birth, the immune system is not yet developed, and the newborn baby is just coming out of an immunosuppressive environment. For the first 3 to 6 months, they have to rely on the IgG antibodies that pass through the placenta from the mother, and IgA antibodies present in breast milk. Gradually, T cells start to mature in the thymus, and B cells in the bone marrow and innate immunity become able to initiate the processes that lead on to acquired responses to infections, and importantly, responding to vaccinations. A fully responsive immune system should have been developed by about 18 months of age. The developing fetus is a partially foreign invader to the pregnant mother, since half its antigens are derived from paternal genes. In order for it to survive, responses to paternal antigens must be suppressed. Some of my own work in pregnant women with autoimmune thyroiditis showed that the regulatory cytokine IL-10 was increased and pro-inflammatory interferon gamma was suppressed during pregnancy. Interestingly, thyroiditis itself improves during the pregnancy, presumably due to immune regulation by IL-10. An important influence on the immune system is high levels of stress, and dips in immune function and increased infections often occur during highly stressful periods throughout life. Again, we found that secretory IgA levels coinciding with increased respiratory illness occurred in students during exam times. In another study of stress in nurses on cancer wards, we showed that interferon gamma was lower than normal and IL-10 higher. Both improved during training for a meditation and martial arts discipline called Qigong, which we were trialling as a potential non-invasive intervention in terminal cancer. These changes were mirrored precisely by reductions in the levels of the stress hormone cortisol. Other interventions that have proved able to mitigate to some extent stressful situations like bereavement, divorce, unemployment, financial problems, and so on, include listening to and performing music, receiving and giving massages, playing sport, taking exercise, communing with nature, and keeping pets. As we get older, the thymus involutes and produces fewer thymocytes and T cells. Furthermore, most mature T cells by the age of 60 will have already been selected for reactivity to antigens encountered throughout life, and few new T cell antigen receptors will be available to react to novel infections. That explains why emerging infectious diseases like coronavirus are such a danger to elderly patients. I'd now like to spend a couple of minutes on how what we eat affects our immune system. Hunger affects far too many citizens of not only developing countries, but in all societies where poorer sectors have an inadequate protein intake. Malnutrition and undernutrition result in measurable changes in the immune system, reduced cell-mediated immunity, reduced phagocyte, NK cell and complement function, cytokine imbalances, reduced production of IgA leading to more respiratory infections, and increased frequency and severity of all infectious diseases. All these are reversible with adequate nutrition. Obesity is an ever-increasing problem in modern society. Amongst the changes seen in the 300 million obese and 750 million overweight people worldwide are smaller thymuses and consequently deficient T lymphocyte numbers and function, deficiencies of T regulatory cells, overproduction of inflammatory cytokines IL-6 and TNF-alpha, and increased incidence of atherosclerosis, diabetes, fatty liver, and asthma. All are reversible with weight reduction. A proper balanced diet will be adequate for normal immune function in healthy people, with little need to use dietary supplements, which can be harmful if taken in too large doses. They may, though, be beneficial in the elderly, in cancer patients, and in elite athletes. The latter have been found to have certain immunological changes caused by overtraining, which can lead to small muscle tears and consequently increased pro-inflammatory cytokines interleukin-1, interleukin-6, and TNF-alpha, which is counteracted by increased T regulatory cells and temporary downregulation of immune responses to pathogens. Vitamins A, B, C, and E, and the minerals chromium, copper, iron, selenium, and zinc are all essential for normal immune function. And antioxidants can reduce damage done by free radicals released, for example, during exposure to atmospheric pollution or intense sports training. Antioxidants are found in some enjoyable treats like red wine, taken in moderation of course, dark chocolate likewise, and fruit and vegetables. Red and processed meat, in contrast, contain high levels of tissue-damaging free radicals. Regular gentle exercise, such as a daily 30-minute walk, is undoubtedly beneficial in maintaining a healthy body, a healthy mind, and a healthy immune system. If not taken to extremes, it can not only improve physical parameters such as blood pressure, blood sugar, and cholesterol, but also relieve stress and depression, improve resistance to respiratory infections, and reduce immune impairment, dementia, and Alzheimer's disease in the elderly. Modern Western living is associated with many practices that diminish immunological health, and these can have extreme consequences. The modern way to bring up young children is to make sure their environment is kept spotlessly clean, and yet exposure at an early age to harmless dirt, bugs, pets, and other children is necessary for the maturation of the immune system. Overuse of antibiotics, especially in the meat and poultry industries, has caused infectious bacteria to become resistant and more difficult for the immune system to remove. Increased stress, reduced exercise, increased pollution, all have damaging effects on immunity. The consequences are reduced immunological balances leading to allergy, cancer, autoimmunity, and infectious diseases, especially the emerging zoonotic infectious diseases like AIDS, SARS, MERS, Ebola, and COVID, all caused by animal viruses becoming adapted to infection of humans. Thank you for your attention. I shall leave you now with my thoughts on how current immunology research might eventually improve control of the diseases caused or influenced by the immune system. I have mentioned the importance of T regulator cells in immunological balance, and what can happen if they either fail to reduce unwanted reactions or aberrantly suppress desirable ones. There is still much to learn about how these cells operate, but I do believe we will come to understand how to induce antigen-specific Tregs in autoimmunity and allergy, and stop them interfering in cancer and infectious diseases. Thank you for your attention.